Thanks for the introduction. Hello, everybody. I hope the audio is good. If not, please inform the host. Otherwise, I'll get started. So you should all be seeing my slides. We will talk about applying Schrödinger's FEP Plus tools beyond the main applications they are generally used for, which is relative affinity calculations employed in lead optimization. And that's, of course, a topic that we have already given a few webinars on. And to give you the idea of what we're about to do today, is I'll briefly have to reintroduce the, I would say, standard or main application of such free energy calculations, which is how do we do relative binding free energy calculations. That's, of course, a scheme that we've shown many times in many conferences and talks. So there is a process that we're interested in, the binding of a ligand to a very schematically drawn receptor here. There may also be a second ligand that we're interested in, and we want to compare those two with the idea to come up with interesting modifications of those ligands to improve their potency. Now, the way this is done computationally is then the main trick in free energy calculations is that we do a non-physical transformation between those two molecules on the horizontal axis here, avoiding the very difficult to simulate physical process on the vertical axis. And since the thermodynamic cycle has to close, we know that the relative binding free energies of those two ligands, so a comparison of which one is a better inhibitor, can be obtained by doing the ligand-to-ligand transformation both in the solvated and the bound state. This is fairly well established, used in many thousand predictions all over the drug design world by now. What I want to talk about today is a little bit of a closer look at the standard thermodynamic cycle that we always show at the very start of an FEP presentation. And the point here is that this standard cycle is one way to set up free energy calculations, but other ones can easily be imagined. And maybe just in general, to understand the cycle, the only two things we need is we need two generally system states or environments, which in the case of binding free energy calculations is a solvated state of a molecule in water and a receptor bound state of the same molecule bound to a receptor binding pocket, itself again solvated, of course. Now, two different environments that we would like to compare, and the comparison here, of course, is related to a binding free energy of those molecules, and comparisons between other environments will be related to other free energies, as we'll see in a second. The second thing we need in that cycle is two different chemical states. In this case, this is one molecule or another similar molecule, and these are the chemical states that we will perform the alchemical perturbations in between. The main limitation of the technology, of course, is that it is a perturbative technique, so the chemical states cannot be too dissimilar. So in our case of two small molecules, we're typically talking about small chemical modifications, introducing a few not too large, not too complex functional groups, or changing a few scaffold atoms for a core hopping approach. But these are really the only two things that we need to have a sensible thermodynamic cycle for free energy calculations. And just to give you one completely different example, here I have drawn another thermodynamic cycle in which our two states now are molecules dissolved in water and molecules dissolved in octanol, and the chemical transformation is still transforming one molecule into the other while it is in water or in octanol. Now, the free energy on the vertical axis here is a transfer free energy between water and octanol. So what this thermodynamic cycle would give us is a molecular mechanics force field-based predictor for log P. And to give you just some basic results here, this is for a series of a few dozen of drug-like molecules. You see that the FEP predicted octanol water partition coefficient correlates reasonably well with the literature data. As usual, there is a correlation, but the function is noisy. There's a few outliers. There's a few deviations. We could look for more complex reasons for that. It's actually not that simple to build a proper model of octanol with a molecular mechanics force field, because it is a long and floppy amphoteric molecule that tends to add some water to itself under experimental conditions. So probably a lot of things could be improved in this prediction protocol. But of course, there's no real interest to do that, because despite our force field log P predictor working, there are of course much simpler, much faster empirical models which also give you log P predictions of about the same quality. So there's probably no point in doing this specific kind of prediction with an atomistic model. But in general, it's nice to see that the approach is robust enough to give you sensible data as long as you write down a sensible thermodynamic cycle. To have a look at a more drug design relevant application, let's talk first about setting this up to predict ligand selectivities instead of potencies. Now, we again have a slightly different thermodynamic cycle than before, but now you'll see there's no more solvated state. We have a state of a ligand bound to a receptor and of a receptor without a ligand bound. And you can see the chemical change now, depicted by the color change, is changing the receptor itself. So what we're now doing is we're simulating a mutation of the receptor in question. In our example cases, this will be a single amino acid point mutation. As before, the chemical change cannot be too large, so we're limited to the number of point mutations that are studied here. A single one typically would work best. And this way of setting it up, the delta delta G predicted now is a delta delta G of selectivity. So it would tell me if the ligand would prefer the yellowish or the green receptor. And that, of course, could be used while I'm doing affinity predictions. On one hand, I could also predict the selectivity of new compounds to close off targets, assuming that the off target is closely enough related to the receptor, to my main target, that a few amino acid point mutations would cover the change. I would like to introduce the test case that I'll show you here is Abl kinase, a protein involved in as a cancer target in a specific type of leukemia. And over the last, say, 20 years or so, a series of inhibitor drugs of Abl kinase have come out, and these continue to be refined over the years, because as it turns out, Abl kinase often shows up with some specific point mutations in tumors, and these are distributed all over the active site, and it's clear that we can assume that if an amino acid residue in contact with a bound ligand is mutated, that could of course have a big effect on the activity, on the efficacy of that ligand in inhibiting the kinase. And trying to understand these processes on a molecular level, we could of course just go on a qualitative level, look at how a specific bound inhibitor, this is imatinib from an X-ray crystal structure from the PDB, we could check what residues does it interact with. So clearly mutations in these residues might affect the ligand binding. And if I look at a different molecule, so imatinib has a pi stacking interaction with some p-loop residues here, while a next generation inhibitor, dasatinib, avoids these interactions with the p-loop. So presumably it is less susceptible to mutations in this position and should remain a potent ligand for p-loop mutants of the Abl kinase. Of course, ideally, we'd go away from such a qualitative picture and would be able to use FEP+ to quantitatively predict the effect of such resistance mutation in the protein on the various ligands that are bound to it. And that is exactly what we're trying to do. And for that, I will briefly swap to our Maestro interface to demo our ligand selectivity GUI, and I've prepared a little example project here. This is a crystal structure of now the dasatinib inhibitor bound to Abl kinase. It went through the standard protein preparation steps that we always undergo, had its force field parameters derived by our force field builder GUI. So we arrive at the final prepared model system of the Abl kinase receptor and the dasatinib ligand bound to it in a reasonable binding pose, this time from X-ray, but could also come from docking or other modeling techniques. As usual, free energy calculations require a properly prepared system with a reasonable binding mode of the ligand already determined. And at this point, we could start studying the effect of single point mutations on the binding affinity of that molecule. And for that, I would open the ligand selectivity GUI by going to Tasks and then Browse under our FEP Plus tools. And you can see, besides the FEP Plus GUI, which is used for lead optimization, there's quite a few alternate applications that already have their own panels. And in this case, we're going to use the protein mutation for ligand selectivity panel. And when I open that, I will import the structures from the project table. So I need the receptor and the ligand as separate entries. You can see that the GUI is already set up partially to the point of supporting multiple ligands, but at the moment we can only do one at a time. So the ligand is being detected here. As you can see, we get the ligand interaction diagram that would tell us what residues we are interacting with. And in the simple-most case, I could just directly pick a residue here. So clearly, threonine 315 makes an important hydrogen bond here. So let's click on that and pick a mutation in the threonine residue, and I will do the most commonly described in the literature is the famous T315 isoleucine mutation, which I'll introduce by clicking on the isoleucine residue here. This little mutation mini GUI might look familiar to you because it is exactly the same that we're using for our residue scanning tools in the Bioluminate interface. So I could go through all the residues involved here, or restrict myself to the ones that are really in close contact to the ligand, and introduce any kind of mutation that I would like. The only requirement is that our prime prediction tools have to be able to properly place the new amino acid into the structure, which typically is not a problem unless I'm introducing massive steric clashes by the mutation that I'm interested in. So limiting ourselves to the threonine to isoleucine mutation, I would set up my calculation as other FEP tools. Ligand selectivity calculations require a CPU host to control the calculation and a GPU host on which the actual MD simulations are going to be run. So I'm not going to run the calculation, of course, as it would take a bit of time, but that's already everything that needs to be done within the setup. I just need to provide the mutations, and if I have a large list of mutations to study, I can pick all of them in the little mutation picker down here, or I can just provide a text file with the amino acid residues and the new residues to mutate them into as well, if I want to run the calculation from the command line. After running it, there is not a terrible amount of output being generated because after running through the whole FEP protocol, if I'm open the FEP protein mutation analysis GUI and load the output file, the auto.fmp file that calculation generates, the only relevant output that we really need is one single number, and that is the one given here. So I have conducted about 8 mutations, various residues that are described in the literature. And if I order them by the predicted delta G, you see, for example, that the T315I mutation comes out with a very big positive predicted delta G. So this would predict that a lot of ligand potency gets lost if that threonine becomes an isoleucine. As you can see, not a huge amount of things that need to be done in the GUI. The setup and analysis are pretty straightforward, but of course the calculation in between is a relatively expensive series of MD simulations that are conducted. Getting back to my slides. if we look at the results and compare them to experiment, you see that the overall agreement is pretty good. We get an overall root mean square error of 1.3 kcal per mole. We identify the most critical resistance mutation correctly, quantitatively here. And you see there is one outlier, the T315A mutation, which might be caused by that specific mutation triggering some protein rearrangement. As usual with free energy calculations, the assumption is that the overall structure is not perturbed too much by the mutation. That is typically something that is true if you're changing small ligand R groups. Of course, with protein, there's always a question if a specific mutation does not impact the local stability of the protein and trigger some refolding. That's a danger that can always occur, and if we simulate this for a little longer, we see that the outlier starts to improve. So this is probably just an issue of sampling. But even with the outlier in place, the predictions overall are very well correlated to experiment and show a reasonable absolute error. Just to compare this with other prediction tools, here on the left are the FEP predicted versus experimental delta Gs. And on the right, I redid the calculation with MMGBSA. And on first glance, the MMGBSA results look actually not that bad. Reason for that is that some of the points are pretty far out of the scale here. For example, the T315I mutation is predicted at plus 40 kcal per mole, which is on one hand, of course, way off, a much too big prediction. On the other hand, it's still qualitatively correct. I mean, this is a very unfavorable mutation to introduce, but as you can see, the difference between FEP predictions and MMGBSA predictions is that one of them are and aim to be quantitatively correct, and the other ones are more qualitatively correct for ranking individual mutations. Now, of course, you probably won't encounter single amino acid resistance mutations regularly in your work unless you're specifically studying Abl kinase. But a much more common question that can be addressed by the selectivity GUI as well is just how much does a given residue affect ligand binding? So we could do something like computational alanine scanning, in which any given residue of interest is mutated to an alanine or a glycine. So we're basically taking away the side chain, making it unavailable for interactions. And this would give us a delta G of how much does this specific residue contribute to ligand binding. And that can, of course, just be of general interest to understand key parts of a protein binding pocket. It's important to remember that those delta Gs are not necessarily additive. There might be non-additive effects from combining multiple mutations with each other, but for a first approximation, this alanine scanning can be very helpful in understanding bound ligands. Now, for a very different application of FEP is the one where we look at the ligand solvation and solubility. And here I have yet to introduce another thermodynamic cycle where you can see the two environments that we're comparing are now solvated ligands again. And the second one is ligands in vacuum. And then the vertical, the physical process here is a transfer energy between water, solvated state, and vacuum. So basically a free energy of solvation. This process itself is, of course, one that we're pretty rarely interested in, but the change in free energy of solvation can be an interesting addition to understanding protein binding again. So one example where this is pretty clear is the T4 lysozyme toy protein test system, which has a lipophilic cavity that binds a variety of aromatics. And what I've done here for these four small aromatic probes, I have calculated the delta G. So this is relative free energies of binding, and we can see that the phenol, the most polar of these molecules, does not bind to that cavity very well compared to the other ones. Now we can, of course, ask ourselves, why is that? Does the phenol not fit into the binding pocket, or is there some other reason? And redoing that calculation by calculating the relative free energy of solvation for those four little aromatic molecules immediately clarifies that the phenol molecule just has, as would of course be expected, a much higher free energy of solvation than the other ones. So its weak binding affinity is actually not caused by any interactions or missing interactions with the receptor. It is solely caused by the large desolvation penalty that this molecule has to pay. Again, to look at a somewhat more relevant case, a typical example of where these calculations might be useful could be the one here, where, say, we start with a ligand A that has a hydroxyl group in this position, and then the idea one comes up with is, why don't I add this guanidinium group here, which forms a nice and stable bifurcated salt bridge to an aspartate residue nearby. And the conversation might go like this: well, I added a nice salt bridge, so ligand B should be much better. A docking calculation might actually tell me that it's a lot better, because the salt bridge is exactly the kind of polar directional interaction that docking scoring functions are so good at picking up. But if you go through the process of actually making and testing the compound, you might realize that it's actually not potent at all. And the reason for that is, and that is one that, unlike docking, FEP Plus should always implicitly give you in the calculations, because it does consider the solvated state, is that these guanidinium groups might make strong interactions, but of course there is a large desolvation penalty to be paid to move them into a binding pocket, to strip them of their water shell. And that is exactly the process that is easily forgotten if you only look at bound ligand structures. There's always the solvated state to consider as well, and that is one that FEP can give you in a nice and quantitative way. To introduce yet another, it's not exactly a thermodynamic cycle, but process that we can simulate with this is the one shown on the right here, where an amorphous crystal, so many of my ligand molecules, are the first state, and the second state is one molecule in water. So here we have a transfer free energy from a crystalline solid state into water, and then of course would give me a free energy of solubility. And in this case, as you see, it's not a thermodynamic cycle. It would give us the absolute free energy of solubility. So it's no longer comparing molecules to each other, but it's explicitly treating one molecule as a whole. This requires a somewhat different setup from our normal FEP calculations. So in this case, we start by building the amorphous crystal using tools from our material science suite. Then we calculate the free energy of sublimation for the molecule by moving one of the molecules at the surface of this solvated crystal here into vacuum, simultaneously calculating the absolute free energy of solvation by disappearing the whole molecule out of the solvated state, and the sum of those two should give us a delta G solubility. Now, there's no GUI for that to demonstrate yet. It is a novel workflow that is actively used internally by Schrödinger as well. And if you're interested in using it, please approach us. At the moment, it's in a beta state where we're actively testing it in some projects. To go back to things that there already are usable GUIs for, we'll have a look at protein-protein binding FEP, for which the thermodynamic cycle looks very similar to the lead optimization one. So now again, we have a physical process of two proteins binding to each other, and the chemical change that we're simulating is once again a single amino acid point mutation in one of these proteins, and the free energy that we should get out of it is the relative binding free energy for the protein-protein complex. The application of that could, of course, be just to understand the interface of two proteins, understanding which residues are important. Maybe that's why they are evolutionarily conserved. Maybe you're searching for hotspots to interrupt in a protein-protein interaction. But it could, of course, also be genuinely to improve the protein-protein binding affinity. For example, if you're trying to engineer antibody-antigen, the stabilization of the antibody-antigen complex. One quick example here, I have a small test system of two protein fragments binding to each other. The reason that relatively odd systems are used here is that high quality protein-protein binding free energy results from the experimental side are often not that easy to get to. So we're often limited to what I would call toy systems. And in this case here, you can see that for about 100 mutations, we again get a decent correlation between the FEP predictions and the experimental free energies. But you'll also see that the noise and the number of outliers is a little bit larger than you would expect in the lead optimization case. So errors are genuinely above 1 kilocalorie per mole, maybe 1.5 or so. That seems to be the typical amount to expect for protein mutation studies. On the right here, I have a graph from a soon-to-be submitted manuscript that looks at a somewhat larger data set of 600 mutations over various proteins and comes to pretty similar results. We get a noisy but decent correlation between predicted binding free energy changes and experimental binding free energy changes. That is already available as a GUI in Maestro. I don't plan to demo very much about it because the GUI looks exactly the same as the one for the selectivity mutation. For protein FEP, you would import a structure from the project table and then just pick residues to mutate, run the calculation, and analyze them using the same residue mutation analysis GUI, which just gives you the delta delta G associated with every single mutation. Yet another way you can run protein FEP calculations is once more a new thermodynamic cycle, which here studies the folding stability of proteins. And in this case, you see that the physical process is the folding, unfolding of a protein structure. Again, the alchemical transformation will introduce specific point mutations into the protein. And this way of running FEP would give you the change in thermodynamic stability of the protein given that a specific mutation was introduced. So you could either understand evolutionary pressure on proteins. Why are some residues able to be mutated, others are not, because they are important for the folding stability. But you could, of course, also use it to engineer thermostable proteins by specifically picking mutations that increase the folded stability of the protein. One thing that I, of course, have to mention, the unfolded state model, as I don't really know on an atomistic level what an unfolded protein looks like, and there's probably no really good models out there. Our unfolded state model is merely a capped peptide. And I probably don't have to emphasize that this is a highly approximative model of the unfolded state. It assumes that there are no intra-chain interactions in the unfolded protein at all. Probably not true in many cases, but in general it seems to work reasonably well. And once again, let me just show you aggregate results from a large-scale screen of a few hundred protein stability mutations, where again we can see that the FEP predictions correlate reasonably well with experimental stability changes. Noisy function, the red dots are charged amino acids for which the prediction quality is notably worse than for the black neutral substituting side chains of the same charge as before. If you want to see this as an optimization problem where I try to predict mutations that are intended to stabilize the protein, we can have a look at an enrichment curve on the left here, and we can see that while that ROC curve looks a lot worse than you would, for example, expect for a virtual screening, a docking enrichment curve, it works well enough that if you used FEP Plus as a pre-screen to pick the mutations to introduce, you should be able to engineer a thermostable protein somewhat faster than by picking mutations randomly or by eye. So in general, and I'll conclude just with this overview image, there are many, many thermodynamic cycles that I can write down that one can run FEP calculations on, and that should give, in general, sensible results. Some of them might be more applicable to drug design than others, but the nice bit, and this is the good part about doing physics-based modeling, as long as the systems that I'm simulating are reasonable models of the real systems, I should expect to get a proper and useful free energy out of the calculation, as long as the sampling requirements are not too large. And with that, I would like to thank you for your attention, and we can move to the Q&A session now.